Hi everyone, today we are going to see about biological classification, Aristotle's classification. Aristotle was the earliest to attempt a more scientific basis of classification of organisms. He classified plants to trees, shrubs, and herbs and animals into two groups, those with red blood and without red blood. With red blood and without red blood. Linnaeus' two kingdom classifications. Linnaeus 1758 classified organisms into two kingdoms, kingdom Plantae and kingdom Animalia. Drawbacks of two kingdom classifications: prokaryotes, bacteria and cyanobacteria, and eukaryotes, fungi, mosses, ferns, gymnosperms and angiosperms were included under plants based on the presence of cell wall, but they were widely differed in other characteristics. It included the unicellular and multicellular organisms in same group, example Chlamydomonas and Spirogyra, were placed under algae. It did not differentiate between the heterotrophic fungi and the autotrophic green plants. Fungi have chitinous cell wall, while the green plants have cellulosic cell wall. 5 kingdom classification. It proposed by R.H. Whittaker, 1969. 5 kingdom classification. It is proposed by R.H. Whittaker, 1969. It includes Monera, Protista, fungi, Plantae and Animalia. This is based on cell structure, thallus organization, mode of nutrition, reproduction and phylogenetic relationships. 3-domain system. It divides kingdom Monera into 2 domains. Eukaryotic kingdoms are included in 3rd domain, thus it is 6 kingdom classification. Characteristics of the 5 kingdoms: Monera, Protista, fungi, Plantae, Animalia. Monera. Cell type prokaryotic, cell wall non-cellulosic, nuclear membrane absent, body organization cellular, mode of nutrition autotrophic nutrition and heterotrophic nutrition. Protista. Cell type eukaryotic, cell wall present in some, nuclear membrane present, body organization cellular, mode of nutrition autotrophic and heterotrophic. Fungi. Cell type eukaryotic, cell wall present, nuclear membrane present, body organization multicellular, loose tissue, mode of nutrition heterotrophic. Plantae. Cell type eukaryotic, cell wall present, nuclear membrane present, body organization tissue or organ, mode of nutrition autotrophic. Animalia. Cell type eukaryotic, cell wall absent, nuclear membrane present, body organization tissue or organ system, mode of nutrition heterotrophic. Kingdom Monera. Bacteria. Bacteria are the most abundant microorganisms. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow and deep oceans. Many are parasites. Based on shape, bacteria are 4 types: coccus, spherical, bacillus, rod-shaped, vibrio, comma-shaped and spirillum, spiral-shaped. Some bacteria are autotrophic, synthesize food from inorganic substrates. Majority are heterotrophs. They do not synthesize food but depend on other organisms or on dead organic matter for food. Archaebacteria. They live in harshest habitats such as extreme salty areas, halophiles, hot springs, thermoxylophiles, and marshy areas, methanogens. Archaebacteria have a different cell wall structure for their survival in extreme conditions. Methanogens are present in the guts of ruminant animals, cows, buffaloes, etc. They produce methane biogas from the dung of these animals. Eubacteria. True bacteria. They have a rigid cell wall and flagella if motile. They include autotrophs and heterotrophs. Photosynthetic autotrophs, example cyanobacteria. They have chlorophyll A similar to green plants. Cyanobacteria, blue-green algae or unicellular, colonial and filamentous, marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of them fix atmospheric nitrogen in specialized cells, heterocytes. Example, nostac and anabena. Chemosynthetic autotrophs. They oxidize inorganic substances such as nitrates and ammonia and use the related energy for ATP production. They help in recycling nutrients like nitrogen, phosphorus, iron and sulfur. Heterotrophic bacteria. They are the most abundant in nature. The majority are important decomposers. Impacts of heterotrophic bacteria on human affairs. They are used to make curd from milk, production of antibodies, fixing nitrogen in legume roots, etc. Some are pathogens causing diseases. Example, cholera, typhoid, tetanus and Cyrus canker. Reproduction in bacteria. Bacteria reproduce mainly by fission. Under unfavorable conditions, they produce spores. Kingdom Protista. It includes single-celled eukaryotes. The cell contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists are primarily aquatic. It is the link with plants, animals and fungi. They reproduce asexual and sexually, cell division and zygotic formation. Protista includes gyrospikes, lionoflagellates, euglenoids, slime molds and protozoans. Chrysophytes, found in freshwater and marine environments, microscopic and float passively in water currents. Most of them are photosynthetic. It includes diatoms and golden algae. Diatoms, they have siliceous cell walls forming two thin overlapped shells which fit together as a soap box. The cell wall deposit of diatoms over billions of years in their habitat is known as diatomaceous earth. This is used in polishing, filtration of oils and syrups. Diatoms are the chief producer in the oceans. Dinoflagellates, mostly marine and photosynthetic. They appear yellow, green, brown, blue or red based on the main pigments present in their cells. The cell wall has stiff cellular plates on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between their wall plates. Red dinoflagellates. Example, gonioxlex undergo rapid multiplication so that red dinoflagellates undergo rapid multiplication so that the sea appears red. They release toxins that kill marine animals like fishes. Euglenoids, mainly freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle. It makes their body flexible. They have two flagella, a short and a long one. They are photosynthetic in the presence of sunlight. In the absence of sunlight, they behave like heterotrophs by predating on smaller organisms. The pigments are identical to those in higher plants. Example, Euglena. Slime molds. They are saprophytic protists. The body moves along decaying twigs and leaves engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium. It may spread over several feet. Under unfavorable conditions, plasmodium differentiates and forms fruiting bodies bearing spores. At the tip, spores have true walls. They are highly resistant and survive for many years. Spores are dispersed by air. Protozoans. They are heterotrophs, predator or parasites. They are the primitive relative of animals. There are four major groups of protozoans: amoeboid protozoans, flagellator protozoans, ciliator protozoans, saprozoans. Amoeboid protozoans. They live in fresh water, seawater or moist soil. They move, capture prey by putting out pseudopodia, false feet. Example, amoeba marine forms have silica shells on their surface. Some of them are parasites. Example, it amoeba. Flagellated protozoans. They are free-living or parasites. They have flagella. The parasite forms causes diseases like sleeping sickness. Example, trypanosoma. Ciliator protozoans. They are aquatic, actively moving organisms using thousands of cilia. They have a cavity, gullet, that opens to outside. By the movement of cilia, the water with food enter the gullet. Example, Paramoebia. Sporozoans. They have an infectious spore-like stage in their life cycle. Example, Plasmodium malatrial parasite. Today we have seen the two topics of biological classification. So in the next video we can see about the remaining topics of biological classification. Video like பண்ணுங்க, share பண்ணுங்க, மறக்காம subscribe பண்ணிருங்க. Thank you. Kingdom fungi. It is a unique kingdom of heterotrophic organisms. Fungi are cosmopolitan. They grow in warm and humid places. Example, mold on bread and rotten fruits, mushrooms, toadstools. White spots on mustard leaves are due to a parasitic fungus. Some fungi are the source of antibodies. Example, penicillium. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi cause diseases in plants and animals. Example, wheat rust causing Puccinium. Except yeast, fungi are filamentous. Their bodies consist of thread-like structures called hyphae. The network of hyphae is known as mycelium. Fungal cell wall is made of chitin and polysaccharides. Most fungi are saprophytes, absorb soluble organic matter from dead substances. Some are parasites. Some live as symbionts. Example, lichens, fungi plus algae, mycorrhiza, fungi and roots of higher plants. Reproduction. Vegetative propagation by fragmentation, fission and budding. Asexual reproduction by spores such as conidia, sporangiospores and zoospores. Sexual reproduction by oospores, ascospores and basidiospores. They are produced in distinct structures called fruiting bodies. The sexual cycle involves 3 steps: plasmogamy, choriogamy, meiosis. Plasmogamy, fusion of protoplasm between 2 motile or non-motile gametes. Choriogamy, fusion of 2 nuclei. Meiosis, in zygotes to give haploid spores. When a fungus reproduces sexually, 2 haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of 2 haploid cells immediately results in diploid cells. To end. In Ascomycetes and mycetes, a dikaryotic stage or dikaryophase occurs. Such a condition is called a dikaryon. Later, parental nuclei fuse and the cells become diploid. The fungi form floating bodies in which reduction division occurs, leading to formation of haploid spores. Based on morphology of mycelium, mode of spores formation and fruiting bodies, fungi are classified into different classes: Phycomycetes, Ascomycetes, Basidiomycetes, Deuteromycetes. Example: Mucorrhizophus, bread mold, and albugo parasitic fungi on mustard. Ascomycetes, sac fungi. They are unicellular. Example: yeast, Saccharomycetes, or multicellular. Example: penicillium. Mycelium is branched and separate. They are saprophytic, decomposers, parasitic, and coprophilous, growing on dung. Asexual reproduction by Conidia produced exogenously on the special mycelium called conidiospores. Conidia germinate to produce mycelium. Sexual reproduction by ascospores produced endogenously in sac-like asci. The asci are arranged to form fruiting bodies called ascocarps. Example: Aspergillus, claviceps, and Neurospora. Neurospora is used in biochemical and genetic work. Morels and truffles are edible. Basidiomycetes includes mushrooms, bracket fungi, or puff balls. They grow in soil and tree stumps and in living plant bodies as parasites. Example: rust and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation in common. The sex organ are absent, but plasmogamy occurs by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which gives rise to basidium. Choreogamy and meiosis take place in basidium, producing 4 basidiospores exogenously. Basidia are arranged in fruiting bodies, basidiocarps. Example: agaricus mushroom, Puccinia rust fungus. Deuteromycetes, commonly known as imperfect fungi because only their asexual or vegetative phases are known. When perfect sexual stages were discovered, they were often moved to Ascomycetes or Basidiomycetes. It is also possible that asexual and vegetative stage have been given one name placing under Deuteromycetes and the sexual stage another name placing under another class. When the linkage were established, the fungi were correctly identified and moved out of Deuteromycetes. They reproduce only by asexual spores. Gonidia. The mycelium is septate and branched. Some are saprophytes or parasites. Majority are decomposers of litter and help in mineral cycle. Example: Alternaria, Trichoderma. Kingdom Plantae. Plant kingdom. Plants are eukaryotic chlorophyll containing organisms with cellulosic cell wall. Some are partial heterotrophs. Example: insectivorous plants like bladderworm and Venus flytrap, or parasites. Example: Cuscuta. Plantae includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Life cycle of plant has two phases: diploid saprophytic and haploid gamophytic. These phases alternate with each other. This is called alternation of generation among different plant groups. Length of the haploid and diploid phases is varied. Also, these phases are free living or dependent on others. Kingdom Animalia. Animal kingdom. Animals are multicellular, heterotrophic, eukaryotic organisms without cell wall. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves as glycogen or fat. Their mode of nutrition is holozoic, by ingestion of food. They have a definite growth pattern and growth into adults that have a definite shape and size. Higher forms show sensory and neuromotor mechanisms. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Viruses, viroids, prions, lichens. In the 5-kingdom classification, acellular organisms, viruses, viroids and prions and lichens are not mentioned. Viruses are non-cellular and truly living, so they are not included in 5-kingdom classification. They take over the machinery of the host cell to replicate themselves, killing the host. Louis Pasteur gave the name virus, means venom or poisonous fluid. D.J. Ivanovsky in 1892 discovered virus. He recognized certain microbes that cause mosaic disease of tobacco. They were smaller than bacteria because they passed through bacteria-proof filters. M.W. Bijarnik in 1898 demonstrated that the extract of the infected tobacco plants cause infections in healthy plants and called the fluid as contagion bivum fluidum, infectious living fluid. W. M. Stanley in 1935 showed that viruses could be crystallized and crystals consist largely of a protein called capsid. A virus is a genetic material, RNA or DNA. The genetic material is infectious. No virus contains both RNA and DNA. Generally, plant viruses have single standard RNA. Animal viruses have either single or double standard RNA or double standard DNA. Bacteriophages, viruses that infect bacteria, usually have double standard DNA. The capsid, made of small subunits, capsomerase, protects nucleic acid. Capsomers are arranged in helical or polyhedral geometric forms. Viruses cause diseases like mumps, smallpox, herpes, influenza and AIDS. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and wing clearing, draping and stunted growth. Viroid, it is an infectious agent with a free low molecule weight RNA and no protein code. These are smaller than viruses. It is discovered by T.O. Dinner in 1971. He found that it caused potato spindle tuber disease. Prions. These are abnormally folded proteins that cause some infectious neurological diseases. These are similar in size to viruses. They cause bovine spongiform encephalopathy, BSE or mad cow disease in cattle, and its analogous variant Creutzfeldt disease CJD in humans. Lichens. Lichens are symbiotic association, mutually useful association between algae and fungi. The algal component is called phycobiont autotrophic and fungal component is mycobiont heterotrophic. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrients and water for its partner. Lichens are very good pollution indicators. They do not grow in polluted area. Okay, today we have completed biological classification. I have uploaded the biological classification chapter into 3 parts. Okay, so go and check it. Video like பண்ணுங்க, share பண்ணுங்க, மறக்காம subscribe பண்ணிருங்க. Thank you.